This meeting is now being recorded.
So hello and welcome to today's RSS PSI Journal Club. We on the topic of adaptive signature designs. Today's meeting is sponsored by Wiley. My name is Stephen Gilmore. I'm chairing today's meeting, and I'm Professor of Statistics at King's College London, and I also chair the RSS Journal Webinar Advisory Group. I'm particularly interested in the area of design of experiments. I'm not an expert in today's topic, so I hope that other people have questions to ask the speakers. So today we have two talks, and we'll hear the presentations first of all from Ziwei Zhang and Gu Ni, who have both published recently on adaptive signature designs, Ziwei in the Journal of the Royal Statistical Society and Gu in the Pharmaceutical Statistics Journal. And then after the presentations, we will have Richard Simon, who is well known as the founder of the inventor of this type of design, who will lead the discussion. And then after that, the floor will be open for questions from the audience. So please, if you do have a question, remember to unmute your microphone to ask the question, and then mute it again afterwards using the *6. Alternatively, you can use the chat facility. So there's a Q&A button at the top of the Global Meet page, and you can use that to type any questions you have, and I will then read these out to everybody listening if you have any questions you want to raise, if there are any difficulties with hearing people, for example, or if you would like further clarification of anything which is said. So we'll move on quickly to the first two, the presentations. So in total, the presentations should take us about 1 hour, and that will leave approximately 25 minutes for discussion and questions. So the first presentation is from Ziwei Zhang. Dr. Ziwei Zhang is Associate Professor of Biostatistics at the University of California, Riverside. He received his PhD in biostatistics from the University of Pittsburgh in 2004, and prior to his current position, he worked at the US Food and Drug Administration as a mathematical statistician, and at the US National Institutes of Health as an investigator in biostatistics. Dr. Zhang has done methodological research in many areas of biostatistics, including causal inference, precision medicine, and clinical trial design and analysis. Today, his presentation is on subgroup selection in adaptive signature designs of confirmatory clinical trials. So I'll hand over to Ziwei now to give his presentation.
Thank you, Dr. Gilmore, for the introduction, and thank you all for calling in. I should also thank the Royal Statistical Society and the statisticians in pharmaceutical industry for organizing this joint webinar and for including my paper in the webinar. My paper is mainly about subgroup selection in adaptive signature design, and it also addresses the related treatment effect estimation problem. So this paper is joint work with my former colleagues at FDA, Meijun Li, Annie Lin, and Greg Soon, as well as my academic collaborator Tom Green at Utah and Changyu Shen at Harvard. I should emphasize that the paper represents our own professional views and not the official position of the FDA. So on the next slide is the outline of my talk. I'll begin with an introduction to adaptive signature designs, and then I'll formulate the problem and describe the proposed methods for subgroup selection as well as treatment effect estimation. I'll show you two examples to illustrate the proposed method and finish the talk with a brief summary. So on slide number 3, it is well recognized by now that treatment effects can be heterogeneous. That is, the same treatment can have different effects on different patients. Sometimes a clinically meaningful treatment benefit is only available to a subpopulation of patients. There are different ways to incorporate this consideration into the design of a confirmatory clinical trial. If a promising subpopulation is known at the design stage, this knowledge can be used to plan a subgroup analysis in a traditional broad eligibility trial or to restrict enrollment in a targeted trial, depending on how confident we are about the subpopulation. Such a predefined subpopulation is often unknown at the design stage. Instead, we may only have a vague idea about a possible subpopulation based on a collection of basic covariates, such as biomarkers, demographics, and clinical measurements. In that case, a target subpopulation can be developed in a data-driven manner, either at the end of a broad eligibility trial or at an interim analysis with the possibility of restricting subsequent enrollment. These designs are known as adaptive signature and enrollment and enrichment designs, respectively. So on the next slide, number 4, I believe, our focus in this work is on treatment subgroup selection in adaptive signature design, and we also consider treatment effect estimation for the selected subgroup. Dr. Simon and co-authors have proposed subgroup selection procedures for specific situations, and here we consider the problem of finding or approximating the optimal subgroup. Our objective is to maximize the power for detecting a positive treatment effect in the selected subgroup, or more generally, the expected gain based on a specified utility function. The latter formulation allows investigators to take into account the size of the selected subgroup as well as the clinical value of demonstrating treatment efficacy for the selected subgroup based on prognosis and available treatment options. Our objective here is notably different from the well-studied problem of finding an optimal treatment regime that maximizes the expected outcome for the entire population. We recognize the importance of that objective, and we believe that it's also important, especially in regulatory settings, to successfully demonstrate treatment efficacy for some, if not all, patients. So on the next slide, number 5, our approach to this problem is based on a simple and general characterization of the optimal subgroup. For a binary outcome, this characterization takes the form of a half space in terms of covariate-specific response rates in both treatment groups and a utility function, if specified. Motivated by this characterization, we propose a subgroup selection procedure which consists of 3 steps. So first, we estimate the covariate-specific response rate in each treatment group. Then we estimate the expected gain for each candidate half space defined by a vector of coefficients and the estimates from step 1. And finally, we choose the half space with the largest estimate of the expected gain. Step 1 is a standard prediction problem. Step 2 can be cast as a missing data problem, for which we adopt an augmented inverse probability weighting approach. Step 3 is a low-dimensional maximization problem, which can be solved using standard techniques. In estimating the treatment effect for the chosen subgroup, a cross-validation approach can be used to remove or reduce any selection bias that may have resulted from the subgroup selection process. And a bootstrap procedure can be used to make inference about the treatment effect for the chosen subgroup. So on the next slide, number 6, an adaptive signature design, or ASD, is just an all-comer study with a prospective plan for testing treatment efficacy for a data-driven subgroup of patients. Here, data-driven is the key. Unlike other adaptive designs, an ASD does not necessarily involve an interim analysis. It is adaptive in the selection of patients for a possible subgroup analysis. Usually, the analysis of an ASD starts with a test of overall treatment efficacy for the entire on the entire study cohort at level alpha 1, say. If the overall test result is significant, then the treatment is considered efficacious for the entire study population. If the test result is not significant, a subgroup is then selected using a pre-specified procedure, and another test of treatment efficacy is performed on the selected subgroup, say at level alpha 2. If the second test result is significant, the treatment is considered efficacious for the selected subpopulation. Here, the significance levels alpha 1 and alpha 2 should be chosen prospectively in a manner that controls the family-wide error rate. So that's a standard ASD. Now, here in this work, we restrict attention to ASDs without a test of overall treatment efficacy. And I'll explain later why we choose to do that. So our analysis plan differs from a standard ASD in that we perform only one test of treatment efficacy for a selected subpopulation, which could be the entire population. So on the next slide, number 7, we introduce some notation here so we can be a bit more formal. So T is a randomized treatment, which is 1 for experimental and 0 for control. Y is a clinical outcome of primary interest, and X is a vector of baseline covariates. The covariate vector X may be used to choose a subgroup for testing treatment efficacy. The question is how to choose such a subgroup and test the associated hypothesis using the trial data. Here, script X is the support of X, and a subgroup of patients is identified with a subset A of script X. If the investigator is only concerned about power, then we want to maximize this power function, which gives the power for demonstrating treatment efficacy for a subset A. There may be additional considerations concerning the size and content of A. Obviously, it's more desirable to demonstrate treatment efficacy for a larger subgroup than for a smaller one. Moreover, successful demonstration of treatment efficacy is more important for a subgroup of patients with no alternative treatments than for a subgroup with many treatment options, right? So on the next slide, number 8, such considerations can be accommodated using a utility function U. So for each value of X, U of X represents the clinical value of demonstrating treatment efficacy for patients with the given value of X. With a slight abuse of notation, we write U of A for the integral of U over A with respect to the distribution of X. This is the realized gain associated with a significant test result for a subset A. For a specified utility function, an optimal choice of A may be defined as a maximizer of this expected gain, gamma of A, which is just the product of U of A with the power function. So that's what we want to maximize in the ideal world. In reality, the exact power may be difficult to calculate, and we work with an approximation based on asymptotic normality. For example, if the outcome is binary, then the exact power is a binomial tail probability, and the approximate power is a normal tail probability, which is easier to calculate. So our goal here is to find a subset A that maximizes G of A and estimate the treatment effect for the chosen subset. Okay, now I can finally go back and explain why we don't have a test of overall treatment efficacy. It seems to me the main motivation for an overall test is the larger gain in demonstrating treatment efficacy for the entire population as opposed to a strict subpopulation. But it's unnecessary now because we have already taken U of A into consideration in maximizing gamma of A or G of A. So this formulation allows us to focus on one test for one selected subpopulation, which could be the entire population. So the next slide, number 9, shows the subgroup selection procedure. Here, to fix ideas, we assume Y is a binary indicator of treatment success and write PT of X for the success probability of a patient with covariate value X under treatment T. We assume that X has at least one continuous component and that the functions U, P0, and P1 are continuous in the continuous components of X. Well, if X is all discrete, then the problem is easier because we have a finite number of subsets of strict X. So at least in principle, we could enumerate all possible subsets. It is the continuous case that is more difficult to deal with, and that's what we consider here. So under appropriate conditions, we find that an optimal subset can be expressed in this form, using this expression here, where the coefficient vector C depends on the subset but not on X. So the subset defined by this expression is really a half space in terms of the utility function and the covariate specific treatment success rates. If the utility function is constant, or if there is no utility function, so we just want to maximize the power, the same characterization applies after removing U of X from this expression. Now, this expression doesn't really describe the optimal subset explicitly because the coefficients depend on the subset. However, it does suggest a practical approach for estimating or approximating the optimal subset, as we explain on the next slide, number 10, yes. So by analogy, we consider a class of subsets of a similar form, where the true success rates are replaced by estimates based on a working model, and the coefficient vector C is arbitrary but has norm 1 for uniqueness. Here, we don't need to worry about any collinearity in X because we only need the predicted values. Collinearity may be a problem with estimation, but not with prediction. For each subset in this class, we estimate the expected gain by substituting estimates of these quantities into the target function G. If you remember, f of A here is the probability of the subset A under the distribution of X, and U of A is the integral of U over A with respect to the distribution of X. These quantities can be estimated by substituting the empirical distribution of X for the true distribution. Similarly, PT of A can be estimated using a sample proportion, but this empirical estimator may be inefficient because it doesn't incorporate covariate information. That information can be incorporated into an augmented estimator, shown here, with a additional term that involves PT hat of X. If you're familiar with the IPW and AIPW approaches to missing data and causal inference, you recognize the empirical estimator as an IPW estimator and the augmented estimator as an AIPW estimator. Although PT hat of X is based on a working model, which may be incorrect, the augmented estimator remains consistent even if the working model is incorrect. If the working model is correct, then the augmented estimator is semi-parametric efficient. Now, on slide number 11, our proposal here is to estimate the optimal subset by a subset in our working class with the largest estimate of the expected gain. The working class is indexed by the coefficient vector C, ranging over the unit sphere. The effective dimension of C is typically 2 or 3, so this is a low-dimensional maximization problem which can be solved using standard techniques. If the working model for PT of X is correct, then PT hat of X is consistent for PT of X, and we can expect C hat and A hat to approach their population counterparts in large samples, right? If the working model is incorrect, then PT hat of X may become inconsistent, and the chosen subset in that case estimates a local optimum, the best subset within the working class. So we get some optimality anyway, and the quality of the chosen subset depends on the quality of the working model. It is actually possible to detect a severe departure of A hat from the true optimal subset by comparing C hat with C of A hat under a parametric bootstrap approach, and the details are given in the paper. So now that we have chosen a subset, the next question is how to estimate the treatment effect for the chosen subset, and this is described on the next slide, number 12. So if P hat is consistent and asymptotically normal for each fixed subset, and assuming some uniformity and continuity, we can show that PT hat remains consistent and asymptotically normal for the chosen subset, which is a random subset. So for asymptotic inference, we don't have to worry about the fact that the same data are used for subgroup selection and for treatment effect estimation. However, in finite samples, there can be a substantial selection bias due to double use of the data, which is a well-known problem. The selection bias can be removed or reduced using a cross-validation procedure, which is described on the next slide, number 13. First, we partition the study cohort randomly into a specified number of subsamples that are roughly equal in size. Each subsample will be used as a validation sample, and the other subsamples will be combined into a training sample. From the training sample, we choose a subset using the exact same approach that we use for the whole sample. Next, we apply the chosen subset to the validation sample and estimate treatment success probabilities. Now, this estimate here is free of selection bias because the two components, PT hat and A hat, are based on independent data, as indicated by their superscripts. And that's the whole point of cross-validation. So we repeat this procedure for each subsample as the validation sample and average the resulting estimates into a final cross-validated estimate. To make inference about the treatment effect for the chosen subset, we use a non-parametric bootstrap procedure that repeats all steps in subgroup selection and cross-validation for each bootstrap sample of n subjects. All right, so that's the methodology. And I'll show you two examples to illustrate the methodology. The first example is shown on slide number 14. And this is the MAGIC study, which looked at the effect of supplemental administration of intravenous magnesium on short-term mortality in high-risk patients with ST elevation myocardial infarction. The MAGIC study enrolled over 6,000 patients and randomized them in a 1-to-1 ratio to magnesium sulfate or matching placebo. Patients in both groups also received standard care. The primary endpoint was all-cause mortality within 30 days of randomization. turns out the observed mortality rate was 15.3% in the magnesium group and 15.2% in the placebo group, with an odds ratio of 1.0. So there's no significant treatment effect. And furthermore, no benefit or harm of magnesium was observed in 8 pre-specified and 15 exploratory subgroup analyses. On slide number 15, in our retrospective analysis, the baseline covariate vector includes age, gender, and well-known risk factors in cardiology. 3 subjects with missing covariate data are excluded from our analysis. We estimate PT of X under a logistic regression model and estimate PT of A using an augmented estimator. Here we want to test for superiority at level 0.025, one-sided. We use a constant utility function, so the utility of a subset is just the size of the subset. The maximization is a 2-dimensional grid search, and the treatment effect estimation is based on 20-fold cross-validation. All right. So from this analysis, we get a subgroup together with estimates of its relevant attributes, shown on slide number 16. Specifically, we look at the size of the total subset, the approximate power, and the expected gain, which is just size times power. We also look at the survival rates for the 2 treatments, as well as their difference, which is the basis for superiority testing. For each quantity, the table shows a naive estimate without cross-validation, as well as a cross-validated estimate. As you can see, the naive estimates are generally more optimistic than the cross-validated estimates. The table also shows nonparametric bootstrap standard errors based on 1,000 bootstrap samples, which can be used for testing treatment efficacy. Before cross-validation, if we work with the naive estimate, the treatment effect is significantly greater than 0. After cross-validation, it's not significant at all. So our analysis of the MAGIC study data is consistent with the previous analysis in that no significant treatment benefit is found in any subgroup. On the next slide, number 17, is the second example, the HEMO study, which evaluated the effect of the dose of dialysis and the level of flux on mortality and morbidity among patients undergoing maintenance hemodialysis. The HEMO study enrolled 1,846 patients and randomized them according to a 2x2 factorial design with a 1-to-1 ratio for standard versus high dose and for low flux versus high flux. The primary endpoint was time to death from any cause, which was not significantly influenced by the dose or flux assignment. However, possible interactions were identified in a post hoc manner between dose and sex and between flux and prior years of dialysis. On slide number 18, the corresponding subgroup analysis suggested that women might benefit from a high dose of dialysis, and that patients with longer history of dialysis might benefit from high flux. Definitive answers to these questions would require a prospective analysis plan, which is impossible in this study because we can't go back in time. So here we present a retrospective analysis mainly to illustrate the proposed methodology. In this illustration, the treatment of interest is the level of flux, and the baseline covariate vector consists of the same covariates pre-specified for subgroup analysis and included in a regression analysis for the primary endpoint. We work with survival status at 3 years post-randomization as the outcome variable. For this outcome to be observed, the subject must have been randomized at least 3 years before the administrative end of the study, right? So we restrict our analysis to this subset of subjects based on randomization date. These subjects happen to have complete covariate treatment and outcome data. Our analysis of this example is similar to the previous one, except for a higher significance level and tenfold cross-validation. And the results are summarized on the next slide in the same format as in the previous table. Here, the naive estimate of the treatment difference in the selected subpopulation is 5.6%, which is significantly greater than 0. After cross-validation, the treatment difference estimate decreases to 3.4%, and the associated standard error increases. But the difference is still significantly greater than 0. So if this analysis had been planned prospectively, it would have successfully demonstrated the benefit of high flux for the selected subpopulation. And here is a summary on the next slide, next and final slide, number 20. So in summary, this work provides some new insights and methods for adaptive signature design, including a simple characterization of the optimal subgroup, a 3-step procedure for subgroup selection, and a cross-validation procedure for treatment effect estimation. The proposed approach has several advantages. First, we don't need to perform 2 separate tests and split alpha. Second, we don't need to worry about collinearity in X because we only need the predicted values. Our approach is also able to incorporate covariate information and improve precision using an AIPW estimator. Although we have focused on a binary outcome in this talk, the main ideas generalize easily to other types of outcome variables. So that concludes my talk. Thank you.
Okay, thank you very much, Ziwei. We will now move straight on, and discussion and questions will be left till the end. So the second speaker today is Dr. Guy Mu, who is a research scientist at Eli Lilly and Company in Indianapolis, Indiana. Dr. Ni received his PhD in statistics from Oregon State University in June 2014, and he has been a clinical and biomarker statistician at Lilly Oncology with hands-on experience of oncology trials. He's been actively involved in trial designs, statistical methodology development in oncology, biomarker data analysis, and regulatory activities. He's been serving as an expert reviewer for 8 peer-reviewed journals covering topics such as biopharmaceutical statistics, clinical trials, and bioinformatics. And today, Gu is going to talk about his paper, Enhancement of the Adaptive Signature Design for Learning and Confirming in a Single Pivotal Trial. So over to you, Gu, please.
Thank you very much, Professor Gilmore, for the introduction. And thanks everyone for joining this webinar. It's my honor to present our research work titled The Enhancement of the Adaptive Signature Design for Learning and Confirming in a Single Pivotal Trial. On page 2, here's an outline of the talk. So I will first provide some background on the biomarker-driven trial designs, followed by a more detailed introduction of the two-stage adaptive signature design, or ASD for short, the focus of the current research, and the objectives. Part of the simulation results will be shown, and some practical considerations will be discussed. I will conclude my talk by outlining some future works. On page 3, a manuscript of the same title was published in the Journal of Pharmaceutical Statistics in May this year, and here's the link to the published paper. The contents in the paper and the presentation today are solely the responsibility of the author and do not necessarily represent the official views of Eli Lilly and Company or FDA. On page 4, So in the past decade, with our increased understanding of the cancer biology, it now becomes a routine to incorporate novel biomarker plans in the hope that we can achieve better identification of the target population. So a targeted therapy may be only beneficial to a subset of the overall population, and we hope that such patient population can be identified using a well-validated biomarker. But in reality, we actually face several challenges. For instance, we may not fully understand the target pathway. We may have very limited data for the preclinical models or early phase studies. The biomarker that can be used for selecting the sensitive patients is not available at the planning stage of a phase 3 trial, and so on and so forth. It is not uncommon to see very promising post-hoc subgroup analysis for large phase 3 trials, some of which may be inactive in the ITT population, but such exploratory analysis cannot support the registration, and a new study with the enriched population is needed to confirm. So the question is there a type of design that within a single pivotal trial, both the overall and subgroup treatment effects can be tested, while the overall type 1 error is still maintained and we have potential power gain? The answer is yes. The adaptive signature design proposed by Friendly and Simon in 2005 is one of such designs. Actually, such two-stage design is not uncommon in practice. One example is the well-known Keynote-001 study in which patients are assigned to either a learned set or a confirmed set so that the sponsor is able to define and validate the PD-L1 biomarker associated with the likelihood of clinical benefit. On page 5. So here are some more details on the two-stage ASD, which consists of 3 components. First, a pre-specified partition of the overall population called the learned set is used to develop a classifier based on which the patients who had superior treatment effect can be identified. We'll call them the marker-positive patients. So this is the learned stage. The remaining patients not in the learned set are included in the confirmed set. Next is the confirmed stage, where we have 2 pre-specified tests for the treatment effect. One test is done for the all-comer, both marker-positive and marker-negative, and the other for marker-positive patients only, where those marker-positive patients are selected for the confirmed set. It should be emphasized that when we identify those marker-positive patients at this confirmed stage, no patient in the learned set should be used. The final analysis involves 2 tests, one on the all-comers at significant level alpha-1 and the other on the marker-positive patients in the confirmed stage at significant level alpha-2, where we normally set alpha-1 plus alpha-2 at 0.05, the overall significance level. So the study is positive if either of the tests is positive. So as you see, there are several key decisions to be made. Authors from the original paper proposed the equal allocation of the patients in the learned and confirmed sets. Although some research shows that when the aim is to minimize the mean square error of the prediction accuracy estimate, one rule of thumb is to assign 2/3 of data to the training and 1/3 to the validation. So there is a key constraint for this two-stage design. The number of patients in the confirmed set needs to be large enough in order to have enough power to detecting the treatment effect. Another factor is the split of alpha between the 2 tests. The authors proposed 80% of alpha allocated to testing on the all-comers, while the remaining 20% allocated to the marker-positive patients. So we have such practical questions regarding the optimal parameters to be clarified. On page 6, so we performed extensive simulations with the following objectives. First, we investigated the optimal allocation between the learn and confirm sets, the impact of different alpha allocations for the 2 tests, and the number of candidate biomarkers at the learn stage. Second, we also discussed some practical considerations, such as the 2-stage randomization of patients, the tools that we developed for performing similar simulations before the initiation of a phase 3 trial, Our ultimate goal is to maximize the overall test power across the plausible alternatives. On page 7, here's a summary of the data simulated. We considered 3 sample sizes, but we only show in this presentation the results of sample size 1400, that is 700 patients in each arm. 3 or 10 biomarkers will be considered, but only one true biomarker X1 is used to define the marker positive and marker negative subgroups by a step function of 0.4. The biomarker values follow uniform 0-1 distribution. We consider 3 scenarios for the different predictive effects of moderate, strong, or the strongest in the marker positive subgroup, with the corresponding hazard ratios specified for both subgroups. The overall hazard ratio is fixed at 0.87, and 100 datasets are simulated for each scenario. On page 8, here's a summary of the test implementation and the parameter specifications. We consider 5 learn-confirm allocations of patients for 30% in the learn set and 70% in the confirm set to 70% in the learn set and 30% in the confirm set. We have considered 4 alpha splits for half split for all comers and marker positive to 0.04 to all comers and 0.01 to marker positive, with increasing alpha spans in testing on the all comers. So for simplicity, 3 biomarker cutoffs to identify the marker positive patients were defined by the first quantile, the median, and the third quantile. If any biomarker is less than the cutoff, we assign it 1, otherwise 0. At the learn stage, a Cox proportional hazards model is fitted using a single marker only, with both the main effects and treatment by marker interaction in the equation. The marker with a significant interaction, or the most significant marker in case of multiple significant markers, will be selected. So in this way, it is guaranteed that at the end of the learn stage, only one marker will be identified with a cutoff value. Page 9. In the next 3 slides, I will show the simulation results for sample size 1400, where on the left panel, there is for the 3 biomarkers, and on the right panel we have 10 biomarkers. Each slide will show 1 biomarker cutoff from the first quantile to the third quantiles. So how to read this plot? The y-axis is displays the empirical power, and the 5 learn-confirm patient allocation are shown on the x-axis. The colors are used to distinguish 3 scenarios of different predictive effects for the marker positive subgroup. The line types are used to distinguish the different alpha splits for the test on the all comers. The horizontal dashed lines present the empirical power for the one-stage design, where a single test for the treatment effect among the all comers is performed, and there's no biomarker component. So we would like to first evaluate the impact of learn-confirm patient allocation. So remember, the true biomarker cutoff is set at 0.4 by our data simulator. And since the actual biomarker values are simulated as uniform 0-1, the first quantile at the median are more closer to the true cutoff of 0.4, as shown in the first 2 slides, slide 9 and slide 10. So when the biomarker is approximately correctly selected, as we see in these 2 slides, we see a general decreasing trend of the empirical power as the allocation changes from 30/70% to 70%/30%, indicating that more patients should be allocated to the confirmed set. So this trend is also consistent across different alpha splits and the number of biomarkers. Although with increased number of biomarkers at the learn stage, the powers are reduced, which is as expected. So we'll go to slide 11. I need to point out that this pattern of decreasing power with decreasing allocation of patients in the confirmed set becomes blurred when the biomarker is poorly selected, as shown here. So in our case, if we use the third quantile around 0.75, which is far from the choose of 0.4, then the patient allocation really does not matter. Slide 12. So in summary, with a sample size of 1,400 in a large phase 3 oncology trial, provided that we have a relatively accurate estimate for the biomarker cutoff and a restricted number of biomarkers, it is recommended to allocate 30 to 40% of patients in the learned set and 70 to 60% of patients in the confirmed set to achieve the maximum power. Slide 13, where we will evaluate the impact of different splits of alpha. Let's go back to page 9. So under the small or moderate sample size, and when the biomarker effect is the strongest, as shown in blue, the highest power is generally observed under the even split of alpha, the solid line here, 0.025 for testing the all comers. However, such even splits often will give us the lowest power if the predicted effect is not that strong, as we shown here in green and in red. Let's go to page 13. So we don't see evident pattern that a particular alpha split will dominate across all the situations. With a much larger sample size, like n equals 2100, the power difference becomes negligible. But with a small sample size such as 700, the overall population, the power difference is evident. So in practice, we need to consider how to split alpha for small populations to achieve the maximum power. Go to page 14. As we compare the left panel with the right panel, where the number of biomarker increased from 3 to 10, we see here in slide in page 9, we see the power decreases potentially due to increase the chance of falsely identifying a no predictive marker. However, it is worth pointing out that even as many as 10 markers at the learn stage, the power gain from the two-stage design over the one-stage design is very clear if the biomarker effect is very strong and the biomarker can be correctly identified. So like as shown here in blue and in green. So this is obvious as the two-stage design power curves lying consistently above the corresponding horizontal dashed lines for the one-stage design powers. Let's go to page 15. So here are some numerical comparisons between the two-stage design and conventional one-stage design. So in case we have a very strong biomarker predictive effect, the two-stage design is a no-brainer as it dominates in almost all scenarios. So when the median biomarker cutoff is used and the optimal values of other parameters are selected, the power gain is really substantial regardless of the sample size. However, if the predictive effect is very limited, for example, probably a wrong biomarker was considered, or if the correct biomarker is selected but the cutoff was poorly determined, then the power gain is minimal, or we may even have power loss. This is easy to understand because in the two-stage design we have to use a reduced alpha or alpha 1 for the all-comer test, and the 70 patients in the confirmed stage are incorrectly selected. Go to page 16. So here are some practical considerations when we allocate patients into the learned confirmed sets. So first, no systematic difference between the two sets should be introduced. So the process must be random to ensure both sets are internally balanced with respect to the treatment arm and important prognostic factors. Otherwise, it will be very difficult to interpret the results due to the confounding issue. A two-stage randomization can be made by first randomized patients to a treatment stratified by the key prognostic factors, and then a subsequent randomization is done to allocate patients to learn or confirm that, again stratified by the same prognostic factors and additionally by treatment. So it should be noted that all the procedures must be prospectively described in the protocol and SAP to ensure the data quality. Next page, page 17. So I'd like to point out two aspects here. So first, for the simulations, we learned that the biomarker identification is very critical in the two-stage design. And in reality, this should never be a hurdle since there are numerous methods available there. We can utilize the results for the preclinical models and early phase studies. Existing results from similar phase 3 studies of the same indication or targeting the same pathway can also be considered. And data-driven methods or machine learning techniques are also at hand. So second, the ASD, the two-stage design, has an important practical advantage in terms of assay development at the end of the learn stage, where the sponsor can work the assay refinement, have the regulatory meetings, or partner to develop the diagnostic kit or CDX. It should be noted that every adaptive design comes with pros and cons, not only for the statistical considerations, such as controlled type-1 error rates and powers, but also for the practical considerations, such as the operational complexity and the cost effectiveness. So to facilitate performing the similar simulation studies as in the paper, I wrote an R package called SimASD available at my GitHub page. So it can be used to create the similar empirical power plots as shown previously to help you determine the optimal parameter combinations planned for the actual phase 3 study. On page 18, so here I list some future works to be considered, such as the multiplicity control at the learn stage, the graphical testing, the choice of biomarker identification methods. What if we have correlations among the biomarkers and the multiplicity issues if multi-markers are allowed into the confirmed stage? So all those ideas warrant more research to get clarified. Page 19, I would like to thank the contributors of the manuscript, my colleagues John, Hollings, Adash, and Eric, two reviewers and editor-in-chief of Pharmaceutical Statistics, Dr. Permute, for their insightful suggestions. Here are the key references. So with that, I will conclude my talk. Thank you for your attention. I'd like to take any questions or comments. Thank you.
Okay, thank you very much, Gu. And I'll immediately hand over to Richard Simon to introduce himself and then to lead the discussion.
Okay. Thank you. This is Rich Simon. I have my contact information on this slide. I don't think I'll go through a long introduction except to say I've recently retired from the National Cancer Institute, and so now I have some new contact information. It's a pleasure to really, I have the opportunity to lead the discussion on these two excellent papers and excellent presentations. And I'd like to start with an overview as to why I think this area is very important. I think that phase 3 clinical trials have two major objectives. One is a test of the null hypothesis of no average treatment effect, and the second is to identify an intended use population. And that second objective comes into play whether there's an average treatment effect or not. And standard clinical trials accomplish the first objective very well, but the second objective very poorly. In cases where there is an average treatment effect, we don't do much to try to identify an optimal intended use population. So consequently, we have small average treatment effects that payers may not want to pay for, large number of patients needed to treat for each patient who benefits, and a result that is overly expensive for our medical systems, our national medical systems, because a large number of patients who were in the original eligibility population don't actually benefit from the treatment. And now, particularly in oncology, where we know that our standard diagnostic categories are molecularly heterogeneous, and we use treatments that are molecularly targeted, we can't really expect to have those treatments have much average effect. So whenever possible, sponsors are actually doing trials in which a priori they, based on biological knowledge, they figure out what is the appropriate population to target and then restrict eligibility. But that, as has been pointed out, is not always possible. And estimating the correct intended use population, I think one fundamental factor is we need to recognize that is actually a classification and estimation problem, not a multiple testing problem. We need to get out of this situation where we use hypothesis testing for all problems in statistics, because it leads us to poor procedures. Now, subgroup selection in the adaptive signature designs, the paper by Ziwei and his collaborators, it's similar in many ways to the the cross-validated adaptive signature design that we published in 2010, but there are some important enhancements to that cross-validated adaptive signature design. But I want to focus a little bit on what is the purpose of these so-called. I tend to think of them as predictive classifiers for identifying the intended use population, rather than using the old terminology of subset analysis techniques. With a predictive classifier is a class. We can think of it as a classifier in which we develop using a pre-specified algorithm on a set of data, and it classifies patients not in terms of whether they're going to have good outcome or bad outcome on one of the treatments or the other. It classifies patients as to whether they're likely to benefit from the test treatment or not, relative to the control or not. And so, in that sense, that's what we're looking for. We're not looking for a bunch of p-values for multiple hypothesis tests that we would then have some multiple correction result for and leave us with something that we don't really know what to do with. What we want to come up with is a predictive classifier, one predictive classifier based on the data, and we want to have some valid statistical properties of that predictive classifier, such as an unbiased estimate of the treatment effect of the population that is predicted to benefit from the test treatment, and perhaps a test of whether of the null hypothesis for that selected population. And with the original cross-validated adaptive signature design, it essentially tried to develop that for any algorithm for developing a classifier, a predictive classifier based on the data. The new approach is focused on a particular type of algorithm, one which identifies the subset of the data which will have the highest power for having a subset-specific treatment effect. Now, when we developed the adaptive signature design and the cross-validated adaptive signature design, we were not really interested in how to do how to have a new way of identifying a subset that benefits from a randomized clinical trial. There are, oodles of such methods, and what they all fail on is that they all end with the statement that, well, our estimates of treatment effect are biased and they need to be confirmed. This subset needs to be confirmed in a future clinical trial. Well, is that anybody really going to do that future clinical trial if you have no statement about how believable your results from this clinical trial are? And so what our objectives were to come up with a way of assessing an internally valid way of assessing the treatment effect of a predictive classifier when you're developing the classifier on the same data that you're using for evaluating it. That's what really had not been done before, and originally we did it in a very simple way, breaking up the clinical trial into one portion for training, for developing a predictive classifier, and a second portion for testing that predictive classifier, for simply on the validation part of the data, simply classifying the patients using the predictive classifier that had been developed on the training set, and then to get an unbiased estimate of treatment effect in the subset that was predicted to benefit, and to be able to do a single significance test of that treatment effect in that chosen subset. So we weren't trying to choose a million different subsets. We were re-specifying the problem as one of developing a single predictive classifier and getting internally valid properties of that classifier. I won't go through the details in the interest of time here. There's a fairly subtle difference between the way we use cross-validation and the way Zoué and his collaborators used it. And the way we used it was essentially using a pre-validation idea in which we didn't do things totally separately based on the loops of the cross-validation to de-bias the resubstitution estimate, but rather estimated for each patient in an entire study based on cross-validation as to whether that patient is predicted to benefit or not. But essentially, in both cases, they're based on using cross-validation to de-bias the resubstitution estimate of treatment effect in the selected subpopulation, and that's a major, I think, accomplishment. What it really means is that we can do subset analysis if we adopt this new sort of paradigm for it and have confidence in the result that the treatment effect result is not going to be biased, and we can actually test the significance within that subset. Now, there's also an important difference in the way the treatment effect is tested within that subset between the way we did it originally in the cross-validated adaptive signature design and the way Zoué's paper does it. We used a permutation of the treatment labels, which, as the paper points out, tests a strong null hypothesis of no subset effect of any type. The new approach is using bootstrap sampling, and it claims that it's actually testing a more precise null hypothesis of no treatment effect in the selected subset. And if so, that would be an important advance. I understand the logic behind it, and I think it's probably right, but I would like to see some more evidence about it. That'll be involved in one of the questions I raised for Zoué. So what are the questions I would raise? One is a question of sample size. How does the accurate estimation of the maximum power subset, how does the accuracy of that estimation depend on the sample size of the trial? And I had some concerns that if you're using the bootstrap With non-parametric bootstrap sampling, you may have 2 or 3 instances of a particular case, and if you're then using that bootstrap data to do cross-validation, you could be introducing a bias into the cross-validation. You'd have one instance of the case in the training set and the other in the test set in the cross-validation folds, and so probably all the instances of a case should be in the same fold of the cross-validation, and I was wondering whether that had been done. And finally, does the test of the null treatment effect really apply to the selected subset based on the selected subset based on the full sample, even though with the cross-validation you have a bunch of different predictive biomarker classifiers being developed? I'd like to see sort of that pinned down a little bit more strongly because it is an important result of the paper. For the other paper, which is, I thought, a very nice paper, too, the paper by Gu, I did want to mention something about the splitting of the alpha. We split the alpha in between a test of the overall average treatment effect and a test of subset effect because we felt like clinical trialists would not want to give up the standard test of overall test of average treatment effect, and so we were trying to induce them to get away from the viewing subset analysis as a post hoc kind of thing. I've seen just too many cases where large clinical trials were done and they wound up doing a subset analysis at the end. The obvious subset based on biology came out significant at maybe a 0.02 level. The overall effect was not significant, and everyone discounted the results of the trial because they had not reserved some of their alpha for subset testing in that obvious subset, and therefore the results of the trial were rejected, and no follow-on trial was ever done because these trials can be, as we all know, very expensive. So I don't think that the advice that statisticians have given is don't believe subset findings. I don't think that's adequate advice. We have better methodology than just regarding subset analysis as doing it as a post hoc way without reserving any of the alpha for it. And so I think we need to change as to the way we view it in the kinds of designs we propose. For example, the FDA in their enrichment guidelines encourage, the use of it's either their enrichment guidelines or their adaptive guidelines, and essentially encourage the use of the adaptive signature design. And so I think we need to really take these things sort of seriously. I will point out that although in the original publication we were suggesting using 0.04 for the overall average treatment effect and 0.01 for the subset effect, when we actually went to plan a trial using the adaptive signature design, and I've cited a reference in which we discussed that in advanced prostate cancer, we found that split was sort of just the opposite of what we needed, where the data was getting very thin for doing the subset analysis. This was not the cross-validated version of it, but where we just split it into training and testing, and we really would be better off, we found, having 0.04 for the subset analysis and 0.01 for the overall effect. And I think that's in agreement with a lot of the findings that Gu reports from his simulations. Also, I wanted to mention there's a paper by Kevin Dobbin and myself about optimally splitting cases into training and test sets, not for the adaptive signature design, but for developing and validating a prognostic signature. But many of the principles as to how you should do that split of sample size sort of carry over to the current question of how they should be split. I think this is my final slide. Questions to propose. Maybe I didn't understand it right, but as I understand it, you have continuous covariates, and yet you're using a fixed quantile of these continuous covariates to see which to use as in the training set for developing the predictive biomarker. And why wouldn't one look at all of the possible quantiles and select the one that maybe works best in the training set? Originally, in developing the adaptive signature design, I was looking at how much does it cost, in the sense of how much does the total sample size need to be increased for getting reasonable power for identifying a subset effect, compared to if you were just doing the clinical trial, looking at the average treatment effect and not looking at subsets. And that's just an additional way of quantifying what this approach entails. And finally, I think there's a potential obvious extension that I wanted to just sort of bring up for comment, and that is when we, as was pointed out, when we do the adaptive signature design, it was not really done as an interim analysis in which you would change anything as you go on in the clinical trial. It was really just how you structure your analysis at the end, and to have an adaptively defined subset. But you could do it as an interim analysis and potentially extend accrual, the total size of the trial, so that you'd improve the power for the subset analysis when the interim analysis indicated that the average treatment effect is not promising, but the subset analysis might be. Thank you.
Okay, thank you,
Hello.
Richard. So the floor is now open for questions. So we do have one in the Q&A box from Danielle Sabanes-Bove, who's asked, would the adaptive signature design also be an interesting option if one only has a single continuous biomarker and subsets are defined by different cutoffs of this biomarker?
Who is that a question for?
It's by Danielle Sabanes-Bove. I'm not quite sure who's in it. I think it's for anybody who wants to answer it.
Oh, okay. Could you repeat the question, please?
Sure. Would the adaptive signature design also be an interesting option if one only has a single continuous biomarker and subsets are defined by different cutoffs of this biomarker?
Right. I think even with a single biomarker, and then one can look in either in a training set at what's the optimal, if you want to cut off, what is the optimal cutoff for that biomarker, and then test that in the testing set, or to embed the whole thing into cross-validation, and but your classifier is based on a cut point, and you use the cross-validation to essentially debias the resubstitution estimate of treatment effect in the adaptively defined subset with that adaptively defined cut point. I think a lot of time, I think that's perhaps one of the most practical applications of these ideas, because very often you're forced to select a cut point based on phase 2 data, and the phase 2 data may not have a control group, it may not have the same endpoint, and so, but you're forced to do that because you think the intended use population has to be all randomized patients, and you don't want it to be all randomized patients. So I think we can use things like the adaptive signature design or cross-validated version of it, and then have an analysis in which the data tells you what's the best cut point and or whether there is no cut point effect, whether it's just a continuous type of thing with using de-biased estimates.
Now, perhaps since Richard has asked some questions to the speakers, so perhaps we should go back to the speakers to see if they have any comments about Richard's questions. So first of all, Ziwei, did you want to say anything in response to Richard's questions?
Yes. First, I want to thank Dr. Simon for the insightful comments and thought-provoking questions. First, let me try to answer, or not answer the question. The impact of the sample size. I haven't looked at it in a simulation study, but I would expect a larger sample size to help with the estimation of the optimal subset. So the estimating the optimal subset for any objective function involves first estimating the objective function for each fixed subset and then maximizing the estimated objective function. Now, for each fixed subset, certainly we can estimate the objective function better with a larger sample size. No question about that. Now, in terms of maximizing the estimated objective function, and that's a little more complicated, and there is possibly a selection bias there. But it seems to me that the selection bias is a finite sample issue. It tends to become a lesser issue with a larger sample size. So I would expect the estimation of the optimal subset to become more accurate with a larger sample size, although I haven't verified that in a simulation study. Now, with regard to bootstrap, that's a really good question, and I must say that I'm not smart enough to give a definitive answer to that question. I think a definitive answer would require a rigorous, careful theoretical analysis that justifies the bootstrap procedure, which I don't have. I haven't done that yet. What I have here is just some intuition, some heuristics. So the way I think of it here, I need to do two things. First, I need to have a point estimator that's approximately unbiased for the treatment effect of interest, and then I try to do inference with a valid standard error. And bootstrap, and I use bootstrap for the second purpose. So the way I think of it is just is quite intuitive. I'm basically using
We seem to have lost Ziwei. Yeah, we seem to have lost sound from Ziwei, so I'm sorry about that. And perhaps if he can come back, he can perhaps answer the third question. But in the meantime, perhaps we could go to Gu to see, Gu, if you have any comments on the questions which Richard asked about your presentation.
Yes, so first, thank you, Dr. Simon, for the questions. So would you please go to the slide, last slide, yes. So for the first question, actually for single marker analysis is totally feasible as the cutoff is determined by the data. So we use fixed quantiles in the paper just for simplicity, since our focus of the research is not about the evaluation of any particular biomarker subgroup identification method, but to investigate the recommended patient allocation, alpha splits, etc. So when the biomarker is approximately correctly selected or is basically poorly selected, as shown in the three scenarios we discussed. For the second question, I think Dr. Simon, you mentioned in your 2005 paper that there will be a 7% increase in the sample size for using reduced alpha 0.04 instead of 0.05, assuming the power of, if I remember correctly, 90%. So I think we believe in case of a very strong biomarker effect in the target population, the power gain will far outweigh the cost of the increased sample size. For the third question, just wanted to make sure that I understand correctly. Are we going to only include the marker positive patients after the interim, or the marker negative patients will also be included? Just for clarification.
Yes, that would be part of what you would decide based on the interim analyses. You would give the opportunity either for restricting it to, or not restricting it, but just increasing the total sample size.
Mm-hmm. Yes, I think, thank you for the clarification, Dr. Simon. I think from a methodology perspective, let me first address the, if we only will focus on the enriched population after the interim. I think it's possible to have an interim analysis on patients for the training set, where tests for the treatment effects on both overall population and subgroup are performed to see if a subgroup effect is more pronounced, while the overall is not promising. So the subsequent patient accrual will be for marker-positive patients only. But I think this way may be a little bit too aggressive, as we have no chance of validating the biomarker in a second stage. So only the enriched population will be included after the interim, right? So there's no chance we can ever test the all-comer. Maybe I would consider this approach only if our understanding of the predictive effect of the biomarker is very strong. For the second scenario, if we consider increase the marker positive and also include the all-comer, I would say that this is also a feasible approach, but we need to maybe give more thoughts on the uncertainty for the how we define the promising for the training set, because we have a relatively small the population to test for the subgroup effect, and when we take a look at the hazard ratio and the associated confidence interval to see to evaluate the variability of that estimate to guide us in the next step after the interim. So that are my thoughts.
Thank you. I know the one trial we were trying to plan, it was difficult at the planning stage because you didn't know what the size of the adaptively defined subset was going to be. So in that situation, it's difficult to size the entire trial to have adequate power for the subset question. But at an interim analysis point, you would have more information about that.
Yes, I would consider maybe at the planning stage, instead of some fixed estimate for the treatment effect, we may have some categories like promising, very promising, or less promising for the planning for planning the patient enrollment after the interim. So we may give some range for the hazard ratio, pre-specified hazard ratio, and to see the interim analysis, how it will tell us like which category it falls and what the corresponding decision will make for the accrual.
Thank you.
Mm-hmm. Thank you.
Okay, thank you. Now, I believe Ziwei is back. Ziwei, if we're able to hear you, perhaps you could go back again and
yes,
we lost your answer to Richard's first question.
I got disconnected. Yeah. So let me just quickly go back to that part about bootstrap. So I was just talking intuitively about bootstrap and using the empirical distribution to approximate the true distribution. So when you do bootstrap, and so some subjects we will have multiple copies of some subjects, and so in cross-validation, they don't necessarily go into the same fold. Could that be a problem? I don't know. I mean, I kind of see these subjects as independent subjects who happen to take on the same values of these variables. And so, yeah, for instance, even if we don't do bootstrap, if we have a discrete X, then if we have a large sample, then we could have multiple subjects, truly different subjects with the same covariate values and also values of Y and T, and they don't have to go into the same fold in the cross validation. So that's just how I see it. Now, it's also true that yes, when we do bootstrap, for each bootstrap sample, we could generally we get a different subset. And again, because in my mind, the reason we are doing bootstrap is to approximate the standard error of my point estimate, and this subset is just an intermediate step in obtaining that point estimate. So as long as the bootstrap gives me a valid estimate of the sample standard deviation of the point estimate, I'm not sure that that is a big problem that we get a different we choose a different subset for each bootstrap sample. But again, that's just heuristics. It's not. I don't have a theoretical justification. Finally, just one more minor point about cross validation. Dr. Simon pointed out a very important point about cross validation. We actually did it that way, as Dr. Simon suggested and also described in their paper. We actually did the cross validation the same way, and we mentioned that as a remark in the paper. I didn't mention that in my talk only because I found it a little complicated to explain. I wasn't quite sure how to explain it. I think I wasn't able to describe it as elegantly as Dr. Simon did. So that's why, and that's an unfortunate omission. We should have explained it more carefully.
Thank you.
Okay, Ed. Thank you very much. So we have to wrap up now, I'm afraid. I would like to thank everybody for joining us, for dialing in. We do hope you have found the session useful, and I would encourage you to leave any feedback you have by clicking on the poll button. So at the top of the Global Meet page, there's a poll button where there's two very simple questions which you can use to leave feedback you have about today's meeting. That's helpful for both the PSI and the RSS for planning future sessions. I would just finally like to remind you that both the PSI and the RSS run regular journal webinars, and both of these are open to members of the appropriate organisation and to non-members. And the next PSI Journal Club will be held jointly with the DIA in September. The next RSS Journal Club has not been finalised yet, but will be also in the autumn or the fall of this year. So do look out in the RSS publicity for that. So thank you, everybody, and I hope it's been successful, everybody. And I think that brings the meeting to an end. Thank you. Bye.
Thank you very much, everybody.
Thank you.
Thanks.
Thank you for holding the journal club. It was, I think, a very nice thing.
Thank you.
Okay.